Hello everyone. Welcome to the 10th talk of CAPS talk series. So last week we heard about protein structures, and this week we are starting and going into the protein function themes. So today there'll be talk on protein three-dimensional models, our hidden jewels for function prediction. Over to you, ma'am.
Thank you. Thank you, Shailin. So you might recall when we were discussing the sequence theme, back in early March, we had a topic about how to find nucleotide binding proteins. And then it also had this topic like needle in a haystack, right? And then we talked about both DNA binding and RNA binding proteins. And Preeta had brought up the point about how we can create repositories, mathematical models or hidden Markov models of protein families that we know belong and bind to RNA, right? So these sort of families were gathered from two perspectives. One which we call as the structure-centric perspective, another which is the sequence-centric perspective, depending on where the primary resource was, whether it's derived from protein structural data bank or the sequence data bank. And in fact, what we are going to hear from Preeta today is given that we have very reliable sort of a binary database of such mathematical models, can we start off from mere sequence information of new proteins and find out whether they have the patterns or the sequence imprints to bind to RNA. And in fact, this server called RStrucfam does a little more. And for instance, once there is a confident association of this new protein to any of these family members, which are known to bind to RNA, RStrucfam also allows the user to appreciate the three-dimensional model of the protein of interest so that we can directly sense how the RNA binding might take place. So here is a case of going through protein three-dimensional modeling so that precise cognate RNA binding also can be predicted. And Preeta will explain to you. Preeta, please.
Thank you so much, ma'am, for the very kind introduction and leading this topic as always. Like ma'am said, like ma'am said, the reason why we thought it will be a good exercise for us and which in the end turned out to be a very pleasant surprise for us was RNA binding proteins have a modular organization, and each of the modules, like you see in different colors and shapes here, they are independently capable of binding to different RNAs. So the partners are not decided based on the context, but it's decided based on the domain alone or a combination of domains together in a protein. Which makes us wonder whether they can retain these functions when in combination with other domains. And so like here you see two different domains can be on the same protein, and they can bind to RNAs on the RNA motifs on the same RNA molecule or on different RNA molecules, which can give rise to same or different functions. And like here, this is some example I had also given in one of the previous talks that RNA binding proteins are unique in a way that even if they are present in multiple repeats in the same protein, they can bind to multiple repeating RNA motifs, and that's where this entire idea comes from, that they retain their RNA binding partner and hence the function. So RStrucfam, what it does is, so when you give a mere protein input sequence without any other information to this web server, it will run an HMM scan against our RNA binding protein database, which has both structural RNA binding protein families as well as sequence families, and then assign a structural family to it. The sequence family will only be later used to assign a function and a Go annotation to the protein. So now if the structure is assigned, then we will be able to know what is the cognate RNA partner, because for structural families, we'll know what is the cognate RNA bound to it. And if there is no assignment of a structural family, then we will scan the sequence families and annotate a molecular function or compartment to it. So this is overall what it does. It looks first into the structural part. If it doesn't find a hit, it looks into the sequence part and assigns a function. So this slide might be slightly overwhelming. So I'll first request you to focus on the left-hand side of the slide. So if the HMM-RBP database or our database of all RNA binding protein families is there, and say there is a family in it, family F, where there are two members, protein 1 and protein 2. Protein 1 is all the proteins, RNA binding proteins which belong to this protein 1 family can bind to say two distinct RNAs, which is R1 and R2. And both of them are known in literature or experimentally to perform a function F1. Say there is another member protein in the same structural family, which is known to bind to a single RNA partner, which is R2, and hence perform a function F2. So this is what we know from our existing HMM-RBP database. So now when the user gives a input protein sequence about which he or she has no information, we take that protein sequence, search it against our database, and say we pick up this particular family F, which we were discussing previously. So now what we can say is this particular protein, which the user has searched for, has a domain which is capable of either binding this RNA R1 or R2 from the protein 1 due to similarity with protein 1, or with R2 due to similarity with protein 2. And it can perform the putative functions F1 and F2. And we had validated this with multiple examples, and it proved to be correct. So the laws of protein function can be extended here, that if you bind to a similar partner by virtue of having a similar structure, then the structure function association can be performed. So based on that, we built a user interface. This is a simple user interface. So here you can search for the families directly based on RNA-based keywords or motifs. So this is the unique part which no other database currently has. It is you can directly enter the RNA motif in which you are interested in, and it will show you which all RNA binding families it can bind to. And you can also browse the entire library of RNA binding proteins from this tab, from the browse option. So when you browse it, what you will get is the putative R1, R2, what I was saying, the different RNA partners and their protein partners and their possible Go functions, like the molecular functions, biological process, and the cellular compartment. And what you will also get, which is a very helpful for the user, is the multiple structure-centric sequence alignment, which we do using our in-house method called comparer, about which you have heard. This is a very powerful method which uses structural information to align sequences, and you can also see how possibly your protein can look like based on this alignment. You'll also get a phylogeny and a possible representation of the RNAs that can bind to it. And one more interesting function which we have is directly pasting your FASTA sequence for the protein here, and then submitting this where the HMM scan will run in the background. And you will get a hit something like this. This will tell you what are the putative RNA binding families, the name of the families, what e-value it bound with, which will give the user a hand on which hit they select. And this will tell you the higher the domain score, it will tell you the better is the alignment and the query coverage. And it will give you further details as to what functions it can perform. You can download the alignment with the query sequence, which will help you to further curate the alignment, or you can take it forward from here directly for homology modeling, which is a very useful feature here. So this is what we had, and any further questions now I can take up now or later. And yeah, I think I'll leave it to ma'am from here.
Great. Thank you so much, Preeta. It was a very clear presentation. And actually today, we are going to have 5 stories. And Preeta's RStrucfam was the first of the 5. And the second one is a rather old one. It started, this particular project started almost at the time when our lab was also formed. And in fact, was taken up by my very first PhD student, Parantusha, whom you have also heard before on functional residue clustering. However, yeah, so here the question really is, how do the transforming growth factors bind to their receptors? So that's the basic question. And this was at a time when the structures were not yet available, especially of the bound form. And that is how the growth factor will interact with its receptor. And also there were known to be type 1 receptors and type 2 receptors, depending on the extent of sequence similarity and so on. So apart from the transforming growth factors, we had activin, we had bone morphogenic protein and so on. And then the question really was, can we use computational modeling, docking, and subsequent structural analysis to recognize or predict how these two will interact, maybe the TGF with its receptor. And for this, Parantu started to look at conserved residues, and he found that there are in fact two types of patches on the receptor end. The receptor is shown in this blue color here, and then under the receptor end there are two patches. The patch in the middle, which we called it as the core of the putative binding interface, is more conserved. And the patch surrounding the core, we call it as the rim. And it's very nice to know that in the literature, these are now highly accepted terms. And the rim, whereas it is conserved, it's also class specific. It will be the residues that you see in type 1 receptors will be there only in the type 1 and so on. Suggesting that they provide the specificity to dictate what receptors should interact with what molecules, what TGFs and so on. And not only that, Parantu went ahead and started the docking between the two. And this is now showing at the growth factor end, similar core region and then a rim region. And it looks like it can just zip in nicely. And what was making me a little uncomfortable is, if you can see on the structure here, it seems to bind to the concave end of the receptor. And that I found it a bit uncomfortable. But Parantu was quite convinced. And in fact, he also performed some rigorous analysis using 100 different poses of how these two molecules might interact, and showed and selected only those where you have highest engagement of hydrophobic residues. So all this is going to be proved right many years later, because this sort of interaction, now we have crystal structures, and our modeling has also been shown to be right. And Parantu is going to present his part of the story in the recording that you will hear very soon. Please stay on. Yeah. This particular project was brought to our notice by Professor Vijay Raghavan. Thank you.
Hello, everyone. My name is Parantu Shah, and I am an Associate Director of Bioinformatics at YelhD Suriname Merck KGA at present. Today, I will be presenting my work on structural determinants of TGF-beta family ligand and receptor interactions. This work was done as a part of my master's thesis in collaboration with Christina Marino-Masolji at CAPS Lab. The work was done around 1999 and published in 2001. As Dr. Sadamini introduced, TGF-beta signaling is well characterized in development across species and diseases, including cancer. The ligand dimers, TGF, BMPs, activins, and others, interact with two types of receptor, receptor type 1 and receptor type 2, for their downstream signaling. The patterns of binding ligands to the receptor is very complex and combinatorial, as shown in the table, which was known at that time in 2001. For this study, we asked whether structural information about the ligand and the receptors, in combination with the evolutionary information that's available from datasets like other datasets, can be used for understanding the determinants of binding. We utilize, therefore, then recently published evolutionary trace method, which requires evolutionary information in form of a sequence alignment and known structures. The steps involve identification of subfamily specific conservation and substitution that can be projected onto the 3D structures. to identify interacting residues. This slides here shows the evolutionary trace analysis of the TGF-beta type 2 receptor family. In the left, you have structure-guided sequence alignment, carefully crafted, which is then used to calculate an evolutionary tree for the receptor active domains. The tree is then cut to identify subfamilies. And then this alignment and the subfamily information then actually is used to identify subfamily specific conservation. And this is expected to be the determinants of binding specificity. So the slide here shows residues identified in both the ligand and in the receptor type 2, which are determinants of binding and specificity. The residues identified in this analysis were basically verified by a published report from either deletion or substitution mutagenesis, in some instances at least. As mentioned before, the ET method requires the projection of conserved residues onto the 3D structure. These residues are not expected to be in a linear protein sequence, and therefore their projection on the 3D structure allows us to really understand the binding surfaces. Of course, it is not available in all cases. So apart from the known structure, we basically carried out homology modeling of receptors and ligands in some cases. So here is an example for TGF-beta ligands. You have 3 ligands with known structures. You have 1 morphogenic protein, inhibin and BMP7. In the middle is the Drosophila decapentaplegic protein. This was of interest because NCBS had many Drosophila melanogaster labs. It is easy to see that the TGF-beta ligands show positively charged lysines and arginines in the TGFs, which are basically changed to a different, or basically glutamates and aspartates in the case of the BMPs and inhibins, while the decapentaplegic proteins does not show charges at basically those residues, giving us some idea of how the binding specificity is working. So armed with this information about known or predicted structures, about the residues that are involved in binding or conferring specificity, we carried out molecular docking of a complex of the ligand dimer with its type 1 receptor to a type 2 receptor, because this is the molecule that we were working with. The docking was successfully able to identify the binding mode that is shown in this crystal structure. This work, or this molecular complex, was actually verified when the crystal structure of activin class terminal receptor complex came out in 2019, so about 18 years after publication. And therefore, we can say that we were successfully able to finish the project. So thank you very much for your attention, and we'll take questions.
That was the second story presented by Rantu. So I hope you can see the slide now. Yes, sorry, the third story that we will be presenting is a fungal protein. And the hero of the story is this gene called YelhD. And the fungus is the Aroia lipolytica, and it has been isolated from contaminated oil areas in Mumbai. And as you can imagine, the environment in which the fungus has to thrive is fairly unfriendly and quite harsh. And in order to thrive, it has to be very robust in being able to take nutrients from everywhere and being able to deal with the catabolism of toxic compounds. And this include halogenated alkanes and so on. And the fungus does it very well. However, the application is we can exploit these properties of this fungus to then be used to make its use in biodiesel production, biodegradation of certain toxic alkanes and so on. So that's how this particular lab of Dr. Amita Ravi Kumar's, the Institute of Bioinformatics and Biotechnology in Pune, got interested. And in one of the visits, Amita was talking to us, to me, about this particular gene, YelhD, which we know codes for a protein that belongs to the huge broad superfamily of alpha beta hydrolases. And within alpha beta hydrolases, there are many families, which means each family will catalyze different chemical reactions, right? Lytic reactions, as the name suggests. And these include thioesterases, epoxy hydrolases, haloalkane hydrolases, and also prolyl oligopeptidases, which we had talked about before. However, the question that Amita posed to me was this particular protein behaves sometimes like a family epoxide hydrolase at a certain pH, and at other times, depending on their biochemical assays, they had found that it behaves like a haloalkane hydrolase. So what is it and why is it behaving so differently at different pH? So that's where we participated, and we wanted to provide a structural rationale. And Harini performed homology modeling of the hydrolase, alpha beta hydrolase of this gene. And it turns out, as the story uncovers, and that Harini will present very soon, as it uncovers, it became clear at different pH, the oligomerization state changes. And depending on the oligomerization state, and if it is a lower order oligomer, the functional loop of the epoxide hydrolases is more available. And that's why, and that's for the protein is able to behave like an epoxide hydrolase. On the other hand, if the epoxide hydrolase function specific loops is hidden at the oligomeric interface, then it's going to behave like a haloalkane hydrolase. And this is going to be presented by Harini. Harini, please.
Good afternoon, everyone. I hope you can see my slides. Yeah. So as ma'am mentioned, I'll be talking about this interesting enzyme, which was found in an yeast that grows in oil-polluted marine waters. So since you have the introduction already, I'll go into the work that we did. So what we started to do is with a phylogenetic analysis, where we selected all the known YelhD proteins, that is haloalkane dehalogenase enzyme-like proteins and epoxide hydrolase proteins, and obtained a phylogenetic tree wherein we included the YelhD sequence, which is the sequence of interest from the yeast. And we found that this particular sequence shares sequence identity with both the epoxide hydrolase and the YelhD-like proteins, and it's positioned in the bit in between in the phylogeny between these two classes of enzymes. And from the structure information, as ma'am mentioned, you can see that both the proteins share similar fold and similar structure. So we expected that this enzyme might have a dual activity and it might behave like both YelhD and EH-type proteins. So what they set out to do at IBB is to look at the oligomeric state of the enzyme and the activity of the enzyme. And they found that at zero salt concentration, the enzyme existed in a tetrameric state. And upon increasing the salt concentration, it could form dimer or a monomer. And the interesting part of this was that at a particular pH, that's at pH 8, it would behave as epoxide hydrolase, whereas at pH 4.5, it would behave as a haloalkane dehalogenase, thereby confirming that this enzyme does have a dual function. So what we set out to do is to build a three-dimensional model using in silico homology modeling of the enzyme from the known sequence, and then use the Gram docking protocol to get a dimer and the tetramer model. We then analyze the interface residues of the dimer and the tetramer structure, and also perform few molecular dynamic simulations to understand what structural features are important for the function of the enzyme. We also set out to look at what are the possible ligands that the enzyme could bind to using molecular docking studies. So I'll explain each of this one by one. So this is the model, tetramer model that we obtained, and we did see that there are some specific structural features of the enzyme that are unique and could be important for its function. One of them is a query-specific loop, which means that this particular sequence was seen only in the query, that is only in the yeast protein enzyme that was identified, and we named it as a query-specific loop, and it was seen to be present at the dimer interface. The other loop, which is the epoxide hydrolase-specific loop, which means that this particular region was present in the enzyme of interest and in all epoxide hydrolase enzymes known till now. And this particular region was found to be important at the tetrameric interface. So we went ahead to do molecular dynamic simulations at different pH conditions to see what happens to the structure of this enzyme at these conditions. So we see, interestingly, and as we hypothesized, that at pH 4, the query-specific loop, which is only present in this enzyme and has a YelhD-like function, is very variable at pH 4 during the simulation.
However, at pH 8, we can see that the epoxide hydrolase specific loop has high variability. This further confirms our hypothesis that at pH 4, the enzyme functions as HLD, whereas at pH 8, it functions as EH, and these loops could possibly play a very important role in this particular function. So at IBB, they then looked at the substrate saturation plots using few of the known substrates for these enzymes. One is the phenyl glycidyl ether, another is the benzyl glycidyl ether. They used the racemic mixture of both the R and S isoform of these compounds and the compounds and the isoforms independently. And they saw that the racemic mixture of benzyl glycidyl ether was the one which showed the better substrate saturation plot, and thereby possibly could be the substrate for this enzyme. They also further did enzyme kinetic study where they saw that again the benzyl glycidyl ether isomers had a very low Kn and a high Kcat by Kn, of which the S isomer had the highest Kcat by Kn, thereby confirming that this could be the likely substrate for this enzyme. So what we went ahead to do is to use molecular docking and see whether we are able to understand what are the structural features in this enzyme that could be important for its interaction with this particular isomer of the substrate, which is SBG. So we first did a molecular docking to find the enzyme binding pocket for the substrate, both SBG and RBG isoform. And we know from previous studies and from the structural information on these enzymes that a histidine residue is very important for interaction. And we selected those dock poses where histidine 325 was interacting with the substrate. We then went ahead and did a 10 nanosecond simulation to understand what are the variations in the substrate binding pocket in the enzyme when the substrate is bound. We found that there are water molecules which are also important for interaction at the substrate binding site along with histidine 325. And in SBG, there was one molecule of water in the binding pocket, whereas there were 4 molecules of water in RBG. However, all through the simulation, we found that the SBG would interact with histidine and water for about 8 to 10 nanoseconds, whereas in RBG, the interactions were reduced. This further confirms our hypothesis that SBG is the substrate and possibly the interaction is more stronger in SBG when compared to RBG. So this is in short the study about YelhD enzyme. Thank you and congratulations to CAPS once again.
Thank you, Harini. Well, it's indeed a very fascinating story, isn't it? YelhD behaving like Jekyll and Hyde, sometimes being an epoxide hydrolase and sometimes being dehalogenase. And this also kind of, I hope, showcases the fact that with the availability of a 3-dimensional model, a very serious collaborator, it's possible to go iteratively and begin to understand some of these substrate specificities as well as the mechanism of action. And also, it's nice that collaboration happens so naturally whenever we travel as well. So that was nice. And then 4th and the 5th stories are going to be back to back, and it will be on with the same collaborator. And that is Professor Ramaswamy, who was in INSTEM some years ago. And the story, the 4th story is on getting to understand the oligomeric preferences of a family of enzymes in bacteria, which are deaminases. And in the gram-positive bacteria, these deaminases, which are actually removing the amine group from the substrate and gives rise to glucose 6-phosphate, which you can imagine is so important in the cycle, sugar cycle. It turns out and is solved, its structure is being solved and it remains as a monomer in gram-positive bacteria. On the other hand, in gram-negative bacteria, where there is strong evidence of allosteric regulation, the enzyme is known to exist as a hexamer, and there are lots of examples of both kinds. And just as one could have settled down with this sort of an idea, in Professor Ramaswamy's lab, they saw the structure of of the enzyme from Pastorella multifida, and they noticed that this particular entry in this enzyme is existing as a dimer when the crystal structure was known. And this was known to be quite different because in most of or the known gram-negative bacteria, it exists as a hexamer. So they were going to conclude the story by saying the PM deaminase acts as like an evolutionary link between the monomer that exists in gram-positive bacteria and then the hexamer that normally exists in many gram-negative bacteria. However, as Ramaswamy came over and talked to us and his postdoc Satya as well, they asked us a question whether it is possible to provide a structural rationale for this. And that's when Vikas Tiwari of my lab also got involved. And what we did, what Vikas did is to take the dimer of the PM deaminase and forcibly on the computer generate the hexameric, provide a hexameric environment to the PM deaminase. And then he went ahead to analyze the putative interface, that is the trimeric interface, which is formed between by three different dimers, and then answer the question whether they can survive, whether they are stable enough or not. And PP check clearly informed us that whereas the HI, which is from Haemophilus influenza, the deaminase from that bacteria, which is gram-negative, has very sound PP check energies at the trimeric interface. The forced model, the forced three-dimensional model of the hexameric form of the PM deaminase shows weak interactions or poor interactions, suggesting that that may be the reason why the PM chose to remain as a dimer. So this is the part that Vikas is going to explain from his point of view, from his work experience. So Vikas, please.
Thank you, ma'am. And in this talk, I will be explaining the oligomeric variation in this interesting enzyme called glucosamine 6-phosphate deaminase. So as ma'am mentioned, we wanted to understand if there is a oligomeric difference between the enzyme and from different organisms. And for that, we started, the collaborator started by solving the crystal structure of the deaminase called NAGB from Haemophilus influenzae and Pasteurella multocida. So Pasteurella multocida is also an opportunistic pathogen, and it can cause diseases including pneumonia. And when we solved the crystal structure for the PM NAGB and the HI NAGB, it shows a monomeric structure shows the 7-stranded parallel beta sheet surrounded by the 8 alpha helices, and RMSD between the structure from already solved structures is very less, indicating that the monomers are very similar to each other, and the maximum deviation was less than 1 angstrom. And when you look at the structure closely, we found that the NAGB from the organisms which are in monomeric state, they lack this C-terminus alpha helix, while the ones which form the hexamer, they have this characteristic C-terminus alpha helix, which can be, so basically from the sequence, we can identify whether the sequence will form as a hexameric oligomer or it remain as a monomer. But when we look at the structure of the high NAGB and the PM NAGB, the high NAGB, as expected, forms a hexamer, which is a trimer of dimers with a 3-fold symmetry as shown in the figure. While the PM NAGB, even though it has that characteristic C-terminus helix, it remains as a dimer. So it was very interesting to understand why this PM NAGB doesn't form a hexamer, while the HI NAGB can form a hexamer. And when we superimpose the two structures, the dimeric interfaces are very similar, and also the interactions formed between the dimeric interfaces involve multiple backbone hydrogen bonds. And again, the residues are very conserved between these two sequences from Haemophilus and also from the PM multocida. Further, to establish that the salt structures are also active enzymes, the enzyme kinetic assays were performed, and it was found that both the enzymes, high NAGB and PM NAGB, are active deaminases. Again, to make sure that the hexamer formed in case of high NAGB is not a crystal artifact, but it remains a hexamer in solution as well. The cryo-EM structure was also solved, and again, it verified that the HI-NAGB forms a trimer of dimers, which is a hexameric assembly. Although the resolution was not great as compared to the crystal structure. And one of the reasons was the preferred orientation of this enzyme in solving the cryo-EM structure. So the question remains is that why PM-NAGB is not a hexamer? So to answer this question, we relied on bioinformatic analysis of these structures. So we started by building the hexamer of PM-NAGB. So the dimer was repeated through 3-pole symmetry and a forced hexamer was built. And then we checked the energetic of the dimeric and the trimeric interfaces using an in-house tool called pp-check. Further, we also analyzed the interface residues, and we found that at the trimeric interface, all the residues are conserved between these two organisms, except for these two residues. And we mutated this residue in silico, followed by energy minimization. And also for the mutant hexamer, we calculated the trimeric interface energy as well as the dimeric interface energy. So if we look at the stabilizing energy at the trimer interface between the high NAGB, which is existing as a hexamer, the forced hexamer of the PM-NAGB, and also as a positive control, we chose to NAGB from the E. coli, which again is known to be forming a hexameric oligomer. And we can see that the stabilizing energy of both high NAGB and E. coli NAGB is much better compared to the forced hexamer of the PM-NAGB for all the trimeric interfaces, which explains like the one of the reason could be that the hexamer of PM-NAGB is not energetically stable and therefore it remains or prefers to remain as a dimer. Later, we also checked as the dimeric interface between these NAGB enzymes from multiple organisms. And we can see that the original dimer, which is the dimer of the crystal structure from the PM-NAGB, has very good stabilizing energy, while that from the forced hexamer has very or poor stabilizing energy. So from this energetic analysis, we can conclude that the PM-NAGB remains as a dimer even though the trimeric interfaces are conserved. And the reason is that the PM-NAGB, upon forming a hexamer, has a very poor stabilizing energy. Thank you.
Thank you, Vikas. That was a very clear presentation of story. I think you're able to see the full screen now. Yeah, great. Thank you. So this was going to be the last story for today's seminar. And it is to do with a different enzyme called the deacetylase. And as the name suggests, this particular enzyme catalyzes the removal of an acetyl group. And it turns out that the substrate is that of an acetyl glucosamine 6-phosphate. And this is again from pastoral maltosida. So what is interesting is if in comparison to the previously determined structure from E. coli, this particular enzyme, which we will call as NAG-A, say, the PM-NAG-A also forms a tetramer, and the E. coli NAG-A also forms a tetramer. And please follow the colors. For each subunit, a particular color has been provided. And if we concentrate on the E. coli one, you can see that across the two protomers shown in green and blue, there is a tight interface, meaning better, stronger interactions as measured by pp-check. And that is shown with the red line here in this graph representation. On the other hand, the interactions that happen between two other dimers, say between the protomer shown in green color and the protomer shown in pink color, you can see that the interactions are relatively weaker. So that is the structure of the E. coli NAG-A. And Ramaswamy again had come over, and this is fact, this happened, this story was much before the deaminase story, which is to then ask with the question that both EC NAG-A and PM NAG-A form tetramers. We see them. However, what happens is the orientation, the mode of the dimer formation is not the same. Both of them seem to retain function and the function is not compromised or affected. However, the mode of the dimer formation is rather different between the two proteins, proteins across the two bacteria. So can we understand why this is the case? And it's almost as if here in the E. coli one, there is the gossip going on between the protomer in green and yellow. Instead of staying together as a symmetric sort of a force, there is a four, instead of having a fourfold symmetry as in PMNAG, the green and yellow protomers are coming closer to each other. And what Vikas had done was to follow these interactions more closely using PP check, compare them between the PMNAG, ECNAG, and so on. And in the process, he found something more interesting, which is these newer sort of interactions are also enabled by means of Velcro residues. And these are called as hotspot residues. And they make sure that the interactions are tight and remain. Right, and these are shown schematically now here with purple dots. And so Vikas is going to explain how using three-dimensional modeling, we can begin to objectively understand the various quaternary differences, arrangements between two homologous enzymes. And by the way, homologous enzymes adopting different quaternary arrangements is not at all new. Even at 58% sequence identity, as is the case here, you can already begin to see variations. Some may form monomer, some may form dimer and tetramer, as we saw in the previous stories. And this has been documented for years now by many crystallographers. But what is interesting is using computational tools such as PP check, you can actually trace which residues, replacements may be responsible for this sort of differences. Vikas will explain from his work how this is being understood. Vikas, please.
Thank you, ma'am. I hope my slides are visible in full screen.
Yes.
So in this talk, I will be describing another quaternary variation in the similar class of enzyme called N-acetylglucosamine 6-phosphate deacetylase from Passerella maltosida. So this enzyme is involved in the conversion of sialic acid to the fructose 6-phosphate, which is again used in the glycolysis. So we began by solving the structure of the PMNAG, and the monomer of it shows the two domain architecture. The domain one is the distorted Tim barrel with alternating alpha helix and beta strand, and there are 8 of them. The domain 2, however, shows certain variation among the different organisms. In case of PMNAG, it is a beta sandwich, while in some other organism, it can be a beta barrel as well. And when we superimpose this monomeric structure on the salt structure from the other organisms, again, it shows very little deviation in terms of RMSD, and it goes maximum till 1.5 angstrom only. And although the monomer is shown here, the functional unit of this enzyme is a dimer because the substrate binds at the interface created by these two monomeric units. And this dimer interface is called the tight interface because it varies a large amount of surface area, and it involves alpha 6 and alpha 7 helices, along with a loop which is connecting the alpha 7 and the beta 6. When we look at the residues at the interface, the dimer interface, we find that most of the residues which are involved in the interactions are conserved, at least between the gram-negative bacteria like the E. coli and the Vibrio cholera. And some residues are conserved among different, even among the gram-positive bacteria, including Bacillus activus. So here the conserved residues are shown as red, and the semi-conserved residues are shown as yellow. Next, if we look at the overall quaternary structure of the PMNAG, among different species, it exists as a different oligomeric state. For example, it exists as a dimer in case of Bacillus or yes, Mycobacterium stigmatis, while it exists as a tetramer in case of E. coli or the Vibrio cholera. The dimeric functional unit involves just the tight interface, as was shown in the previous slide. And in case of PMNAG, we found that it exists again as a tetramer, which is a dimer of dimers, but it is not the same as has been reported for the other bacteria, which is, for example, if we compare it with the E. coli, as ma'am suggested, the quaternary organization is different. In terms of that, PMNAG has a dimer which are formed between, say, subunit AB and say a subunit CD, and then the dimer-dimer interactions are very weak. So we call them as a weak interface. While in case of E. coli, the one subunit can interact with all three remaining subunits. And because of this difference in the quaternary organization, there is presence of a solvent channel in case of PMNAG, and which leads to many water-mediated interactions among the subunits. So the take home from this we got is that PMNAG shows unique tetrameric quaternary architecture compared to the other tetrameric NAG. Just to emphasize that this happens even though the sequence similarity is pretty high between these two organisms. If we closely look at the dimer-dimer interface in case of PMNAG, so here we are looking at the weak interface. The very obvious things are the abundance of the water molecule at this weak interface. And these are involved in the multiple water-mediated hydrogen bonding interactions between the corresponding residues. So this is shown for subunit A and C. And other than hydrogen bonding interactions, there are many hydrophobic interactions as well. And another major difference between the E. coli NAG and the PMNAG is the lack of the ionic interface. So ionic interface is formed in case of E. coli tetramer, where multiple salt bridges are also observed, which are not there in case of PMNAG. So to understand the reason behind this quaternary organization, we again relied on the bioinformatic analysis, and we started by predicting the hotspot residues at the interfaces. So the hotspot residues are the ones which give specificity to the interface and also provide the stability. So mutation of these hotspot residues can lead to the dissociation of the subunits. And we could identify residues like tyrosine, glycine, glutamine, and aspartate. Interesting thing which we observed is that among these hotspot residues, only the aspartate 363 was conserved between the PMNAG and the E. coli NAG, while these four hotspot residues were different in case of E. coli NAG. And if we mutate these hotspot residues to their corresponding E. coli residues, it results in very poor stabilization energy. So one of the reasons that it has a different quaternary organization is because of the presence of these varying residues, which are also the hotspot residues. Further, we wanted to check if we mutate these residues individually or in combination, what will be their effect on the structural stability. And we could see that all of these hotspot residues are majorly destabilizing in nature, or they are neutral in nature. And similarly, we also checked the interface energetics, and it shows the similar trend that if we mutate the hotspot residues, the interface gets destabilized. So the reason for it to have a different kind of tetrameric organization is because it has a varying residues on the surface, which are also the hotspot residues, which might lead to organize the subunits in a different way compared to what is there in case of E. coli. So this is what we conclude from this study. Thank you.
That was very nice, Vikas. And you actually kind of also explained how the comparison of evolutionary conserved residues as well as hotspot residues can explain quaternary level differences. And I think that's a very cool approach. On the whole, I hope the audience had enjoyed listening to the 5 stories. First was the RStrucfam web server, which will permit us to predict RNA binding properties to proteins from mere sequence information. And then we talked about Paranthu's experience of modeling the interactions between TGF and its receptor. By the way, Paranthu is here. If anyone wants to ask questions, we can promote him to the panel position. Let's see. I think he's already here as a panelist. Okay. Yes. Oh, great. Yeah. Hello, Paranthu. And that kind of was predictive at that point. And then years ago, the prediction of their interaction turned out to be right. And then the third story was the YelhD where at some pH the enzyme behaves like an epoxide hydrolase, and some other pH it behaves like a YelhD halogenase. And then the fourth and the fifth stories were on deaminase and deacetylases, which were revolving around the quaternary arrangements and their differences between homologous enzymes. So with this, I would like to acknowledge and also mention the people who presented today's talk as to where they are right now. And Rita, for instance, she's in a company called Influence, and she's somewhere in the other part of the Bangalore, and she's been able to come online and present the RStrucfam story. And on the TGF receptor interactions, we had Parantu, who had initially given his recording, and now he's online. And then he is currently a scientific director of bioinformatics, a biomarker, and also in Serrano in the US. And Harini, who was a student before and who had done the YelhD modeling, is currently in Raghu's lab as a postdoc. And the deaminase and the deacetylase stories, as you heard from Vikas, were in collaboration with Ramaswamy's lab. And it was presented by Vikas Tiwari, who is currently a PhD student. With this, I would like to request Shafi to present the list of papers that are relevant to the 5 stories that we had presented today, and also to inform the topic, the title for the next seminar. Shafi. Right. As Shafi is picking up his slide, I will also like to say we had several questions and answers where questions online, and the answers have been provided by the speakers then and there. So if there are any more questions, please do not hesitate. You can just raise your hand and we can permit you to, allow you to ask your question. Thank you.
Hello, everyone. So these are the publications relevant to today's talk, the 4 short stories. First one, RStrucfam, Preeta Ghosh, in the beginning, she talked about this web server. And the second one, Parnisha's paper, TGF receptor. And following to that, Harini presented about YelhD, the collaborative work with IBB Pune. Then Vikas presented deaminase and deacetylase. That's a collaboration with Professor Ramaswamy. So these are the 2 recent papers from those works. And for the next week, in CAPS talk, we have another set of short stories in function theme. When we have titled it "When Protein Modeling Go Hand in Hand with Functional Assays." Please stay tuned.
So for the audience, I want to say that you can not only see the speakers like Preeta, also Parantu has joined online. So Parantu is one whose work we had also talked about when we discussed a function theme on the functional residue clustering, looking at serine proteases, right? So he's here with us, and in fact, he has also worked on another project within his 2 years of stay as an MSc student, which we had not yet presented. It was on inositol triphosphate receptor modeling. So there is Parantu. Hi, Parantu. Hello. Yeah. So it's nice that he has come online. And what time is it for you in the US now?
It's 6:30 in the morning.
Okay, all right. So it's a good way to face the day. I hope the rest of the day also will be very pleasant for you.
Thank you.
So yeah, if there are no further questions, please stay tuned. And as Shafi had announced, the talk, it's going to involve functional assays. So it's going to be even more exciting in some ways. So until then, the audience, please stay safe. We will see you next week, Thursday at 3 p.m. Bye.